Hello, and welcome to Building Better Peppers, connecting your minds to plant breeding. I'm Michael Washburn. I'm the Seed Savers Exchange Preservation Director, and I'll be your host for this session. I'd like to give a special thanks to Alliant Energy for sponsoring this session. I'd like that all participants in the Seed the Future Grow Something Good focus on being respectful, being curious, being patient, and speak from your own experiences. And try to avoid jargon, be conscious of intent versus impact, take space and make space, and take care of self. And by embracing these community agreements, we embrace a collective responsibility to create spaces that are respectful, inclusive, and empowering for all. So we are here today with Bala, Rathinasabapathi. And Bala has a Ph.D. in biology from the University of Saskatchewan. He also has his master's in genetics from Indian Agriculture Research Institute in New Delhi. And he got his bachelor's in agriculture as well. And he's a current professor of horticultural sciences at the University of Florida in Gainesville. Now, today, Bala will be talking about the story of a pepper breeding program where undergraduate students are given opportunities to do hands-on projects to create new cultivars. And attendees will learn how to prioritize plant breeding projects, what it takes to do breeding, and they may also be inspired to take up vegetable breeding as a hobby. Bala, thank you so much for being here.
Thank you, Michael. Thank you for the introduction. Thank you for the opportunity to be here. And I love seed saving, and I think all agriculture starts with seed saving, and it's the core activity in plant breeding. I'm here to share with you something that we do in my teaching program, and I teach for the plant breeding as well as other aspects of plant biology. And this particular project came about 2010, and I'm going to tell you about what I call as building better peppers. Why peppers? I'm interested in peppers. Yeah, I like peppers as an interesting crop, and it's also very important for Florida. So I have here, we named this project Building Better Peppers. That really means that the peppers in the market are not good enough, right? So we got to build it better. So that's one of the things we got to learn when you're plant breeding. That means if you want to improve something, you got to know what doesn't work so that we can fix it and make it better. So that's one of the goals, how to identify a problem, and then we want to teach the students how to do plant breeding. And I call it experiential learning because I give them an opportunity for students to just play around and use their creativity to make something good and better. In that process, they learn a lot of things, not only just growing and making new varieties, but also how to grow all aspects of horticulture. So here we came up with this idea. We have a lot of junk food in the world, and people like a lot of junk food. But can we make vegetables as great as junk food and maybe better? Nutritious, tasty, colorful, really cool to eat. And so, can we then take up projects that will just take one aspect of fruit quality or vegetable quality or something that other people are not researching so that we could research about it and find new things and then make better product out of it. So that's a goal we set out, and the students and myself together, we wrote it out and say we are going to breed tasty, nutritious, colorful, attractive vegetables that outcompetes other food. So I teach a course on vegetable breeding. In that, we are using pepper breeding as a model, and we use a lot of that. However, I'm also involved in breeding eggplant and a few other vegetable crops. So there are three methods to improve crops. One group of method that has been used for a long period of time, hundreds of years, is cross-pollination, an induction of mutation and selection, and this is all under traditional plant breeding. So most of the varieties that you and me will see in the store that comes from traditional breeding, many of the heirloom varieties that we use are traditional breeding in a different way because families saved seeds for their use. The other method is what's called transgenic crop technology, where a gene gets introduced. Again, there are a lot of controversies in that technology, whether that is there. But however, the technology is different from the result, because depending on what gene you put in and who controls it, and then there are aspects to that. Then the last technique is more recent phenomenon, that's genome editing, where genes are tweaked and all of that. In this particular project, we only wanted to focus on traditional plant breeding, simply cross-pollination and selection, and that's what we are using. So the project started out with a small group of students. There are some 15 students that were there. It was a classroom in 2010, and we discussed ideas. At the time, I was using only Johnny seeds and Park seeds, a couple of seed catalogs. But later on, now it has grown to hundreds of seed sources that we are sourcing seeds from. But I asked the students, they can just go and find any pepper they want, and then they can cross breed them and then grow them in a greenhouse. We put a number lock on the greenhouse, and the students can go in and out anytime after I teach a little bit. So they picked, some of them picked that just looked attractive on the catalog. There's a variety called Bulgarian carrot. It's a very sweet name, but it's horribly hot. I mean, it's very hard to use the Bulgarian carrot. I mean, some of you might have grown. And another student took a sweet pepper that was a round of Hungary, and that's a pimento cheese type heirloom variety. But since then, many hundreds of varieties. We used a jalapeno. Many hundreds of varieties were we got the seeds, and then the students grew them, and then they made controlled crosses. So what is a controlled cross? So within a species, you can make a cross and combine characteristics together in a new plant. That itself is a power, because you could actually combine different colors, shape, and quality characteristics and get new combinations. So I usually tell them plant breeding is like a Lego art, like you have multiple colors and multiple shapes, and you bring them together to make a completely new product. So here it's what that's shown, and I'm just going to illustrate it with some pictures. That's a pepper plant, and all of these are the flower buds. But these flower buds are not yet open. But from this, we choose one bud that is going to open tomorrow. That means mature enough bud, ready to open next day. But that previous day, we remove the anthers, the male part of the flower, using a forceps, and very carefully do that so that the female part of the center is not altered or damaged. And after that, we take all of the anthers one at a time. There are like 5 or 6 in peppers. And after taking that, we close it in and carefully leave it so that we don't want any bees pollinated. This is why we say controlled pollination. So we cover it with cotton, a little bit of cotton, and that's just safely there. Next day, when the flowers are opening, and we take another flower that's open flower, that's a male plant from which I want the pollen from, and then I dust the pollen onto the stigma, and very carefully. And after doing that, that flower is thrown away, the male source, and then the whole pollinated flower is covered with a little cotton. And then we put a little tag, and then that tag we write the date and also which plant was used as a pollen source, and leave it there, and we got a cross fruit. That cross fruit will develop and it will mature, and then after maturity we collect the seed from it. That seed is the hybrid seed from which if I grow a plant, I'm going to get a hybrid plant. So this technique was taught, and then the students did these kind of things. Remember here the Bulgarian carrot, and when it's crossed to round of Hungary, you got an F1 that kind of had a shape that was so different from each other, both the parents. Now we are actually creating a new characteristic that was not there in either parent, and that's the most interesting aspect of this game. So not only that, the next thing is, how is this tasting? Is it good? That kind of question. So initially we took very simple idea of just simply using color as a way to study, because color is related to nutrition. I'm just having a few more pictures here. For example, this Islander, this variety has got this purple coloration, and the F1 has got a lot more purple color. We create a lot of different purple color types. Here you can see the mini apple, Cajun bell, and what we are getting as a hybrid is a little shape-wise, it's a very interesting, what I call a top shape of the fruit. So we made this project objective. One of them is we're going to focus on inheritance of interesting traits, in this case, mostly fruit shape, fruit size, pungency, and also fruit color at maturity. And then we will use them so that we will make some novel combination so that we can come up with the new cultivars. So that's a goal. The other thing we were interested in is some of the things that other scientists are not studying. So we looked at some fruit characteristics like antioxidant concentration, pigment and flavor, keeping quality. More recently, we added resistance to diseases and root knot nematodes, which is a problem in Florida. So we are looking at those. So interestingly, every time when we do these experiments, we get to grow hundreds of plants. Sometime this is a visiting scientist from Brazil who was with me for a year, and we grew 300, 400 plants at a time inside a greenhouse. And then the students are taking care of them because we are searching for the novel combination that we want. And then some of the plants are grown in the field. So we have about 10 miles from here. We have a field where we are growing with a plastic mulch and drip irrigation, which is the standard practice here for conventional growing of peppers. Some of the lines we are also testing under organic production condition. Some of them are traditional production condition. This is an undergraduate student, Mackenzie, who is taking care of the plants in this particular field. So I'm going to tell you a little bit of science story. One of them is many of the colors that we see in fruits, particularly one in pepper, are very interesting colors, and they come orange or red, and those colors are all carotenoids. And these carotenoid structures are shown here. What you're seeing as a little squiggly line is a beta-carotene, lycopene, lutein, and zeaxanthin. These four of them are important in our diet, in human nutrition, particularly beta-carotene. Why? Because beta-carotene is important for eyesight and many other things, and it comes from carotenoid. In other words, beta-carotene is coming from carrot, but it's also in pepper and many other plants. So the beta-carotene is broken down in our body to make vitamin A, which is very important for nutritional purpose. So what we analyzed is that we took pepper and many other vegetables, and we kind of rated them based on a score called a nutrient density score. What is a nutrient density score? It's calculated as percentage of daily vitamin requirements, all kinds of minerals and vitamins, and then daily requirement per unit calorie, and then we rank the vegetables. So what you see here, the blue color bar is actually the nutrient density score, and you can see the top one, very first one on the list is the spinach, and the second one is kale, and the third one is a red bell pepper. Now there are multiple different peppers that are shown. That's because there are some peppers that have a really low nutrient density, some other peppers that have a high nutrient density, which meant that nutrient density score can be a character that we can play the game of selecting and making new peppers with a high nutrient density. Does that make sense? The orange color one is showing the carotenoids alone, and you can see the carotenoids are high in certain things like a dandelion, collard green, spinach, and kale, lots of carotenoids. But in peppers, although it's a high source, there is a bit of variation. In other words, peppers they have less carotenoids than, for example, say greens and papaya and mango. So we are also thinking that we can improve the carotenoid amount and composition. So that's one of the research goals that we found. This is actually a composition of carotenoids in a pepper that was from a 2013 study. What you see is the highest amount of carotenoid we find is beta-carotene, which is very important. So beta-carotene is a good source, and that could also be improved, and we are working on that. I mean, eventually we will select for that. The pathway for beta-carotene is pretty complicated. So what's showing here, or what's shown in this figure, is a carotenoid synthesis in red fruit, in pepper fruit, and in peppers accumulate a red color pigment called capsanthin, another one called capsarubin. These two are the brilliant red of the pepper fruit that you see when the pepper become ripe. So I'm just going to tell you one small scientific story where the students participated. 13 students participated in one study and we discovered something interesting scientifically, but also product-wise, we are working on multiple lines that are interesting. We took these two heirloom varieties, Bulgarian carrot and Round of Hungary, made an F1, which is the hybrid, and then we took the seeds of the F1, grew those plants, and then went to the next generation, which is the F2. And the second generation will actually segregate for a lot of characters. I'm quickly going to show you a picture, but before then, I'm going to show you that the Round of Hungary, the red variety, has got a thick, fleshy pericarp, or the fruit wall. At the same time, the Bulgarian carrot has got a very thin pericarp. So one student examined why, and in that case, we found out that the thick pericarp is due to both larger cell size and also more number of cell layers in the pericarp. So you have a bigger cells and more number of cells. That even before doing any kind of genetics, we know there must be two genes controlling it. One controls the cell size, another controls the number of cells. And we may be able to tweak it and get varieties that are interestingly different in pericarp characters. Then we kind of collected data on fruit length. We collected data on pericarp thickness, width, pungency, how hot something is. And then what's called a Brix is a sugar content, so an approximate amount of sugar content. And you can see the Round of Hungary is 5.2, which is equivalent to 5% sucrose. But on the other hand, Bulgarian carrot is only 1.3, which is like 1.3%. Okay. So let's see here. We grew the F2 plants in little containers, and it was in a greenhouse. And you can see the student helping grow these plants. And then these are taken care of, and then we collect fruit from it. So this particular figure is showing one fruit representing one F2 plant, because F2 is a larger family of plants. So every one of them, what you see is that lots of variation. Is really size is different, shape is different, color is different. And so since we were interested in carotenoid and color, I thought, oh, we should just simply select the yellow ones or orange ones, and the orange will represent more carotenoids, actually beta carotene. So we thought, oh, we could just simply select for that. So then one of the issues is that even though the color is apparent and we can group them as yellow and also red, I thought there is more to that color, because some of them have two shades of red color. Some other, all the orange and yellow ones can be also grouped into two. So we measured using a machine the color of every fruit, and then that machine gives color readings, and it's called a Kodak color meter. It's pretty easy to use, and in that we have a A value which represents red color. So higher the A value, more red the color is. In the tomato industry, they usually use 30 A value as a benchmark for the red color. If it's not matching 30 A value, the tomato wouldn't go to the market. That's a standard. So when we analyze the color values, the red color value, what I'm showing you at the very top, you see the little graph, and at the bottom we have a red color space. That's a plus value numbers, and then the y-axis is the yellow color space values. When we look at that, all these little dots on the top, which is not filled circles, they come from yellow, orange fruits, and all the ones that are red are coming at the bottom. So all the ones that are here, that makes in the line. So what we found is some of the yellow ones also had a very high red color value, suggesting there must be a red pigment even in the yellow fruit. We are fooled by nature, because we are thinking that yellow color means there is no red pigment, because the red pigment comes from a yellow precursor. So we thought, oh, we will not have the red pigment. But we found, based on this machine's reading, there must be a red color in it. So we did test that idea using HPLC. And what you're seeing is capsanthin and capsorubin. These two peaks are the red pigment. And that was there in the Bulgarian carrot, the orange fruited variety. And this is the Round of Hungary. Capsanthin, there's a lot more, and than capsorubin. And both of them were present in both varieties. This was news, and it's also news to the world of science, because people, studying carotenoids, they thought the color is a very clear indication. But we know the color is not everything, because the color can be buried in some of the characteristics of the pepper. So this and many other data that we collected from this particular group of plants were published in a journal, and this is a Journal of American Society of Horticulture Science. That's a 2015 study. We have published 3 more work like this, and here it's inheritance of fruit traits in Capsicum annuum, which is the pepper. And then I'm also making another important point, the heirloom cultivars as a source of quality and some of the parameters that are interesting is reported in this paper. Since this particular time, I have kind of made additional lots of work on various different heirloom peppers, and we are making good progress in understanding some of the traits that other people have not yet studied, and we are also making validation for what other people have done. We could see that. So right now, more recently, we are interested in tiny peppers. So we call it a mini pepper project. In that mini pepper project, we are using one variety called sweet pickle. Many of you would have grown this. It's a kind of ornamental pepper, but it has got all this shades of color from one plant. So we are using the sweet pickle as a source for some of the color variants, and we are making lots of tiny size fruit, which is about 10 gram to 20 gram per fruit. And these varieties are leading up to lines that are making this type of color variation and also shape variation. Our goal in this particular project is to make miniature peppers that are easy to eat. So remember, when you go to the store, you can buy mini tomatoes that are like tiny tomatoes. So we think, oh, we have an opportunity here to make the pepper like that, if only we have new colors and cool shapes so that we can attract people to eat more of it. But first of all, you need to be very good. So here is the mean fruit weight. And so in one of the progenies, a student has analyzed the fruit weight, and you can see it varies from 20 to 25 gram per fruit to somewhere like 5 gram per fruit, which are much smaller. So we are into other kind of things that are related to carotenoids, and this one is a mutation to chlorophyll. So there's a variety called a fish, and the fish pepper is a hot pepper that has this kind of streaky character, and we use that genetics to move that sticky character to other peppers, and we have some very interesting looking peppers. We're just calling them gator stripes because everything in Florida is gators. So that's my presentation. This is show you a very small sliver of what we do. I'm training about 20 students a year on plant breeding, and these students, not necessarily they're going to all become plant breeders, but that's not the point. The point is that they appreciate all the hard work that goes into growing vegetables, and also they know that they learn a lot of techniques about germinating seeds and growing transplants, taking care of them, pest control, managing disease, all of those kind of things. So they learn in that process. And very end, there are 1 or 2 students who are really excited, and they will just go out of the way to do things that I had never thought I would do. Like there was a student, 2 students who were interested and said, oh, we are just artists, and we don't want to, but we like your pepper project, but we don't want to do the sciencey stuff. So they designed that little logo. Okay, that was their contribution. So the students come into the class with a 1 credit hour, they will register, and then they have this 1 semester period. At the end of the period, they may not achieve, like 1 semester, they cannot achieve a lot, but they can achieve up to a certain point, and then they will give us the seats and the report. So a next group of students come in and pick it up. So this is what I call a research relay. Like that means a research project never dies because students are taking it further and further in many different directions. So we have many different projects, approximately like 20 of them right now. It's just only a matter of how many I can handle in terms of practically managing them. Most of it is handled by me. In other words, I know which seed comes from where and that kind of pedigree information. And at the same time, we have a couple of grants that came from Florida Department of Agriculture and Consumer, and those kind of grants were given to us on specific projects that relates to root knot nematode resistance and also relate to bacterial spot resistance. Those are problems in Florida. And so we could actually then get some money to work on those. But those projects are very streamlined. That means we have a very specific goal, and that's what we're going to do. But in the student projects, anything goes, and students play with it, and they come up with all kinds of things, ideas. And the idea is to then make students excited about plants, and particularly in this case, vegetables, and also um, ask them to solve problems. And some of the problems are not easy to solve, but students are trying hard. So that's a fun part of it. I enjoy every minute of it. Of course, I'm overwhelmed. I eat sometime a whole day. I eat tiny slivers of pepper because I need to know which pepper is good. And sometimes the hot pepper is not very easy to taste and figure out, because that's you can't just chew the hot pepper. That's hard. But we have everything. We're scoring with a scale of 1 to 5, 1 to 5, and then anything that is interesting, we pull it out. Only thing, I get so excited about various things. Like the other day, I found a pepper that was very bitter. I'm really crazy about why is it bitter? What's in it that makes that particular line bitter? So I saved the seed. I want to push it further. Only thing is that industry people don't care. They would have thrown it long ago and say, "Oh, that's bitter. Forget it." The kind of thing. So I ended the presentation here. Any questions? I'm happy to answer. Thank you.
Yes, Bala. Thank you so much for that. That's truly fascinating. I feel we probably could have went on for quite some time after this. So thanks for really packing that in for us. That was just wonderful. I have a couple of questions myself, but first, uh, Victoria asked, do you know if any other organizations are doing similar work with heirlooms?
Not I know of, specifically heirlooms. I also read the literature on peppers, and I found out that nobody has taken the heirloom as a commodity, as a group of plants to mine for characters in plant breeding. So I thought there is this opportunity exists. Paul Bosland, he is a famous pepper breeder in New Mexico. So he wrote a book on peppers, and in that he mentions that heirlooms are great because they are already adapted to eastern United States. Lots of people are growing it, and then he also mentions that it has not been systematically used in breeding, and that's what made me interested in it and say, well, there is an opportunity here. One of the problem with a very popular crop like pepper is that all of the so many people are interested and so much of work has been done. So we did some work on shape of the peppers, and then we found out a 1938 paper that described what we found. Then scientifically it's not that exciting. So I want to find a new question, new interesting things. So this is where the heirlooms come in. Yeah.
So we've got Tom asking, have you ever used Cubanelle peppers?
Yes, we are growing cubanelles now. Yeah. Sometimes what happens is students are interested in a specific pepper and they bring it to the program. And the cubanelle was brought to me like that. And a couple of different varieties we have grown. Some of them have some wilt problems, so they kind of died. And then we have to identify which one would be good. And we are working with all. So the other thing is that the bell pepper industry is big, and that's what sells most. So most of the programs in the United States that are working with peppers, they're focusing on bell peppers. So we are actually then moving away from bell peppers and focusing on what I call specialty peppers, all sorts of specialty peppers.
I'm going to allow one last question here at the end. So Gabriel's asking, what happens when you initially cross-pollinate an F1 with an heirloom or an F1 with an F1? Is it possible to still dehybridize and stabilize into an open-pollinated new variety?
Yes. Answer is yes, you can do it. And I have a student called Ed Vieira, and he was just wanting to do this type of thing. So he actually crossed multiple F1s back and forth in many different ways. And all the lines that were from that is called a Vieira number, like a Vieira 2, Vieira 3, Vieira 4. And that comes from a complicated mess of a cross. But the nice thing is it's like, putting marbles in a bottle and shaking it, similar like that you're shaking the jeans and everything gets mixed out. Yeah, it's possible.
So, what are you seeing with heat when you're crossing the sweet and the hot peppers? How are you seeing that continue on?
The heat is always more, it's a dominant. So that means it'll show up all the time. You will find recessive sweet ones but it'll be much rarer in the population. We can search for it. The problem is searching for it is hard, because when you cut the fruit, I usually cut the tip of the fruit and then chew on it to figure out whether it's hot or sweet. But then sometime it'll be fooling you, because it feels like you have a sweet pepper, but in reality the heat is near the seed at the very bottom of the fruit. So then the selection is a little difficult. Heat is always a little difficult to work with, because the students are not, if they are not careful, they hurt themselves. That's the problem.
Yeah. Well, Bala, again, I thank you so much for this. It's just fascinating work. I'm sure you'll have some folks now definitely continuing to follow up on the wonderful research you're doing out there with peppers. And yeah, I look forward to see the results and where all this is going. But yeah, just absolutely fascinating, and can't thank you enough for sharing this with our community.
Yeah, thank you. I really appreciate it, and I'm all for seed saving, and I really appreciate all the service you guys do, and thank you for the invitation. I really appreciate Janine and your help, and everybody else here and everybody.
Excellent.
Thank you.
All right, thanks so much.
See you. Thank you. Wonderful.